Immunoelectrophoresis. As the word suggests, this method combines the two separate methods and their principles, electrophoresis along with immunoprecipitation. I think we have dealt in detail with immunoprecipitation and its types. We have talked about that. I will briefly talk about electrophoresis with you all, considering you have some knowledge about it since you have performed agarose gel electrophoresis in your earlier standards. So I will quickly go through electrophoresis through one or two slides, and then we will talk about immunoelectrophoresis. How do we combine them? How do we modify the method? And how do we get the results? So we have these four methods to learn, although there are many more types of immunoelectrophoresis. We have these four types: immunoelectrophoresis, countercurrent immunoelectrophoresis, rocket immunoelectrophoresis, and two-dimensional immunoelectrophoresis, which is also called as crossed immunoelectrophoresis. Let us see them one by one, but as I said, before knowing the immunoelectrophoresis, let us quickly glance through what is electrophoresis. So basically, under the application of electric current, we are separating components from the mixture, and that mixture is generally of nucleic acids or proteins. So when the mixture is DNA, we generally use agarose gel electrophoresis, which I think you all have performed in your first year. Whereas if the mixture is of proteins, we have to impart charges onto the protein. Unlike DNA, which is naturally charged negatively, so it runs towards positive electrode. In proteins, we have to apply charges, we have to impart charges using SDS, and then we make them run along the polyacrylamide gel. We of course run it through the gel, and because of the charges we get the separation. Also because of the molecular weights and their capacity to run through the pores in the gel, we get the separation of the components. We apply the current which is able to pass through the medium. We use buffers for this purpose so that the electric current is carried. So basically requirements are the charged particles and the medium that is capable of carrying a current. And I hope you have the basic idea how do we carry out this. But this is not the lecture to discuss about electrophoresis. So considering that you have a slight idea that we are going to utilize the current to move the charged particles from one end to other. Okay, that much knowledge of electrophoresis is sufficient to understand the immunoelectrophoresis. Let us move ahead and further discuss about what are the factors that actually influence the rate of diffusion of immune components like antigen and antibody into the gel. So first and foremost, its concentration. Second is its physicochemical properties, and third is the gel structure. So generally the gels that we use for immunoprecipitations are either of agar or agarose. Agarose is a purified version, so agarose gels are generally preferred, and 1% soft agarose is what we generally use for immunoprecipitations. After this background information, let us understand the principle of basic immunoelectrophoresis method. So this is a method in which we are going to separate antigens according to their charge. Please understand, antigens being proteins in nature, if we have a mixture of antigens, they will not have any particular charge on them, right? They are proteins, and proteins carry different charges according to the amino acid composition, and therefore we are going to first separate antigens based on their charge. So we are not going to place any antigenic picture near any particular electrode. Okay, we are making them run towards their respective electrode based on their charges. So please understand, in the procedure what we do is we take an antigenic picture and we are not really charging that antigen, right? So let me try making a diagram for you all, although I have a pictorial view that I will show you later. We take a glass slide, right, and we have a gel poured onto a glass slide. So that is, of course, in any electrophoresis, immunoelectrophoresis, that's a preliminary requirement that we have to have a gel, and that gel slab we are going to bore a well. So where exactly should we bore a well? So if we are going to decide that we are going to charge this side of the gel with positive electrode and this with negative electrode, where should we bore the well? Since antigenic is a mixture with different charged antigens, we want to put it near the center, so that whatever charges they are carrying, they can move. Either positively charged will move towards negative electrode, or negatively charged will move towards positive electrode, so on and so forth. Then you would ask me why you have not put the well here, or you have not put the well here, right? I can put it here, fine, I can put it here, but not in the center. Okay, there is a reason for it, because I want to, first of all, I want to utilize the best way this slab portion, and along with, if I remove the, if I remove these parts, what I'm going to do is, after I actually make them run under electric current, after electrophoresis has taken place, I'm going to cut a trough. Now this trough can be cut earlier too, but then it's okay to cut it later too. So on one side, just look at the positions where I'm keeping. Antigenic mixture is here, so antigens are separated on this side, and anti-serum trough is being cut on this side. To this trough, I'm going to add anti-serum. Okay, this anti-serum will contain antibody. Okay, whereas antigen was here in the mixture. So I hope you've got a slight idea about how the procedure is. And after this, we are going to allow natural diffusion of this antibody across the gel. Okay, and after this, we will see R formation wherever you will have precipitin band zone of equivalence created for the antigenic particular component. So if you have not understood my diagram, let us refer to a better picture here. Okay, what they have done now, they have cut two wells. You can do, as I said, you can do in the on the topmost, and you can do in the bottommost, but not in the center. Now, why both sides they have done? So that they can use more and more antigenic pictures for the same trough. And they have cut the trough in the center. So now you can see in this antigenic mixture, you had three components. In this antigenic mixture, there was only one component, right, which moved towards negative electrode. So this was positively charged. This is negatively charged, and this is positively charged. Also, if you understand the, their size will also make them run through the gels at different rates. And this is where you can see the actual arc seen for the each component, right? So this is the pictorial view. This is the actual view on the slide, how you get. the arcs mixing with each other, and you have to actually recognize which arc is for what component and so on. Basically, this is a qualitative method just to detect presence or absence of a particular antigenic component. Okay, and antibody in the serum, antiserum for a particular antigenic component. Okay, if you ask me what is the application of this method, uh, before understanding the application, let us again view the QB diagram of this method. As you can see, the antigen well in the center, the component antigen separated after electrophoresis. Trough was cut even before, that is okay, but antiserum was not added after the second step, after the separation of antigenic mixture. You've added the antiserum which contain antibody which started diffusing, and wherever the zone of equivalence is reached, you will have the precipitin band. Okay, so after this complete procedure, we can also see a video in which it talks about how we actually move on with the process. So a better uh, view of method. We use agarose because it's neutral and it is not going to interfere with the reactions. And what exactly we do is we take agarose gel in a slab, and then we bore a well, antigenic well, right, to which we add a mixture. Trough is also they have bored, but they have not added any anti-sera to it. Electrophoresis is first done in order to separate out antigens. You can see antigens separated on the basis of their charge, and then you add antiserum, and then you allow that antiserum to diffuse, and wherever you will have the zone of equivalence, you will see the arc formation. Again, this is a qualitative method. At the max, you can talk about the relative concentration of the components. For example, let me explain with the help of an example. You can detect the presence of proteins in the serum, or you can check whether the components of the serum are in appropriate amount. Now you'll ask me, you just said that it's not quantitative, then how can we check whether their amount is appropriate or not? That's a good question. We are actually carrying out qualitative estimation only, but based on the arc formation, we can compare and talk about whether the relative concentration is high or low. For example, these are the different proteins present in the plasma. You can see there are number of proteins. So how many different types of arcs can be observed? It's beyond your imagination. But then if you want to see, these are the different arcs that could be formed. And if you know what is the exact arc that albumin should give, or alpha 1 should give, or alpha 2 should give, or beta or gamma should give, then you will be able to probably get those arcs and mark them and note whether they have been following the exact their location as per the healthy uh, immunoelectrophoresis might give healthy sample of antisera might give. So you would come to know whether your antigenic mixture, that is here, for example, the plasma, contains appropriate amount of proteins or not. So this is how we can actually comment about the relative concentration of serum proteins or plasma proteins and qualitatively detect their presence. If you have understood immunoelectrophoresis, let us understand the countercurrent immunoelectrophoresis. Now, when I say countercurrent, what comes to your mind is something that is flowing against the current. So we have two flows actually. We have one in direction in which antigen moves, and in one direction, opposite direction should I say, antibody moves. Okay, so why we are exactly doing this is because we want the faster results. So as I said, we always try to modify the method in order to get a better result. So by doing this, we are making them run towards each other. While in earlier method you saw that after putting antisera in the trough, we had to wait for antibody to get diffused across the gel, and then the arcs might be seen and that would take time to develop and so on and so forth. But applying and making them run under the influence of electric current will help it to form very early. And that's what is the advantage of countercurrent immunoelectrophoresis. However, antigen and antibody are not having any natural charges, so we need to impart charges on them, and that is the most disadvantage of this method, that we are adding up steps of our work, making it more tedious in order to get the faster results. But yet, this method is being currently used for detection of hepatitis B surface antigen as well as detection of bacterial and fungal polysaccharide antigens in cerebrospinal fluid. If you have understood countercurrent immunoelectrophoresis, you would appreciate that both the methods learned so far are qualitative in nature. There is nothing talked about quantitative. So we yet try to modify the immunodiffusion, and we come up with something called as rocket immunoelectrophoresis. So rocket electrophoresis was result of modifications made in 1966 by Laurel, and yet again modification made in 1992 by Erickson and Wyman in a way in order to make a quantitative estimation using uh, immunoelectrophoresis. So why the name rocket is because of the typical type of electrophoretic arcs that is seen, precipitin bands, that the type of arc that is shows, you can see it here, it is typically rocket like, right? So it is the name rocket electrophoresis. So what exactly are we doing? We are having antigen and we are having antibody. But so far in both the techniques of immunoelectrophoresis and countercurrent immunoelectrophoresis, we had two different wells for antigen and antibody. However, for the first time, we are putting antibody in the agarose gel itself. So while setting up the gel, we have to ensure that we are adding antibody to it. Okay, point number 2. When we get antigen, which is commercially available, we are not putting any unknown concentration of antigen. Okay, we are taking antigen in a known concentration and we are imparting charge to it. So second step is to impart charge to the antigen. Third step is we have to dilute the antigen. Now, if you remember, in biochemistry related practicals so far, you have uh, many times you have quantitatively estimated presence of proteins in the sample, unknown sample, say using Follin-Lowry or biurate method, if you have ever done that, or you have quantitatively estimated the concentration of DNA using DPA method or RNA using orsinol method, so on and so forth. There are many, even glucose or dextrose in DNSA method. So there are many methods in which you have actually counted the concentration in an unknown sample using a standard graph, and that is what we are going to use in this method. What we have done is we have taken a known antigen of the known concentration. After giving a charge to it, we have made a serial dilutions of it. And as you can see, here we have got an increasing antigenic concentration. We have bored 6 different wells in the gel. And in each well, we have added antigen in an increasing concentration. And after adding this antigen, as soon as we add, we are going to run it across the gel using electric current. Because our antigen is negatively charged, it will flow towards positive electrode, and then because it is in the flow, the precipitin bands are in the rocket-like appearance. And more the concentration, more is the height of the rocket, right? So the height of the rocket corresponds directly or directly proportional to the concentration of the antigen in the well. And same, by this we are going to plot the standard graph, and in this you can see this is the decreasing concentration of antigen, whereas this is an unknown. You can correspond this, and you can you can extrapolate this height when you plot the standard graph of these four concentrations, and you can tell me what is the concentration of antigen in this well. In this way, we can perform rocket immunoelectrophoresis. Of course, this method too has some advantages, some disadvantages, but still it is applied. The disadvantage could be you have to again charge antigen negatively. Advantage is for the first time we are getting a quantitative estimation with a very simple method, and as I told, height of the cone is directly proportional to the concentration of the antigen. So this method can also be applied to find out the concentration in an unknown sample. Then we move ahead with the last method of immunoelectrophoresis, which is called as two-dimensional immunoelectrophoresis or crossed immunoelectrophoresis. Now this method is little difficult, little I said, because it is not much different from the others, but it is little difficult to imagine. Although if you have learned and if you have understood the first three methods correctly, you will realize the simplicity of this method and modification of the immunoelectrophoresis done in this method very easily. So basically, two dimensions, meaning we are going to run electrophoresis twice. We are going to do it twice. First in one direction and second in some other direction. But at the same time, you can see it is also called as crossed immunoelectrophoresis, meaning the paths of the two electrophoresis are going to cross, right? And they are going to cross at 90 degrees. So let me tell you what exactly we are doing. This method is again a quantitative estimation for the composition of antigenic mixture. And as I said, we are going to run electrophoresis in two different dimensions. In first, we are going to separate the protein mixture as we did in the immunoelectrophoresis, but there is a slight difference in this that I'll also tell you. And second dimension, we are going to run it across like antibody containing gel, like a rocket electrophoresis. Okay, so this picture will help me to explain this. Antigenic well, as you can see, it is bowed towards negative electrode. So when we take antigenic mixture, it has to be negatively charged. Okay, so charging of the antigen is important in this method. So except for the first immunoelectrophoresis, we are imparting charges every time in all the methods. In countercurrent immunoelectrophoresis, we imparted charges on both antigen as well as antibody. But in crossed immunoelectrophoresis or rocket immunoelectrophoresis, we are giving charge only to antigen. Then what we are exactly doing after this is we are running the antigenic components across positive electrode under the electric field. This is the first dimension. And then we want to keep this gel at a right angle, right? This we kept it at a right angle. So we have a mixture separated, and since the antigens are negatively charged, they will run in this gel as well. This gel contains antibody, and while running, they will give us the rocket-like precipitin bands. So this is the second dimension of immunoelectrophoresis, and one advantage of this over rocket immunoelectrophoresis is that in rocket we used only one antigen and we made it diluted, right? In different wells we use the different dilutions. But in this particular gel, in this particular technique, we are using a mixture, we are separating it, and yet we are able to quantify. So this is a very important modification where we are actually mixing all the immunoelectrophoresis and rocket immunoelectrophoresis principles together, and this is how we get the bands seen It requires an experience to understand and calculate each and every arc, observe it with an eye, so you can see how faint the first arc is, right? And how small the ninth arc is. But then, yes, it requires expertise, and that's why uh, you will require some years of experience for understanding the results. And these are some other methods like tandem crossed electrophoresis, intermediate gel crossed, or fused rocket crossed. You can find out about these methods if you are curious about immunoelectrophoresis. But for our syllabus, I think this much, I mean, these four methods are sufficient for you all to understand. I hope you have understood immunoelectrophoresis and its types. If you have any doubts, comments are always welcome. Thank you.